following podcast was taken from a webinar presented by Dr. Joe McFadden from Cornell University titled Methyl Donor Nutrition in the Transition Dairy Cow. To view the full webinar and access slides referenced during this podcast, visit balchem.com/realscience and use the search bar to jump down to this webinar presented on May 4th of 2020.
Welcome, everyone. Please take note of my email in case you want to reach out to me after. I can direct your attention to any one of the three meta-analyses at the bottom of this slide. I think what we have a general appreciation for rumen-protected choline feeding in early lactation and the benefits that can result. The practice has consistently demonstrated increases in milk yield and energy-corrected milk yield. The effect is certainly more pronounced when cows are fed diets low in metabolizable methionine. It is estimated that approximately 50% of this response is due to increases in dry matter intake. However, endogenous mechanisms, which we really don't fully understand quite yet, explain the other half. Choline feeding also supports increases in milk fat and protein yield to explain gains in energy-corrected milk. It also supports the ability of rumen-protected choline to minimize body condition loss in early lactation and reduce the incidence of postpartum diseases. I think this is great news. I'm sure you think the same as well, and something that we have heard before about rumen-protected choline feeding. But this is not your average choline talk. So I'm going to present new information about choline biology in the dairy cow. I will cover three distinct topics that will each end with a conclusion. For topic one, my study has recently focused on fatty acid nutrition. And I'm going to make a case for why we should consider fatty acid feeding when we feed methyl donors like choline, but also methionine to cows. For topic number two, we will discuss choline degradation in the rumen, but also the intestines, and focus our attention on the role of trimethylamine oxide and its potential effects on health. And lastly, for topic number three, we have a series of trials in development studying a complex lipid called lysophosphatidylcholine. You may recognize this as lysolecithin. I'll make a case for why we should consider this lysophospholipid as an immunomodulator in fresh cows, but also pre-weaned calves. Overall, my goal is to talk to you about something new, something that you haven't heard about before. But please realize, I think we are certainly at the forefront of the science, and there's much more research to do.
New research is changing everything we thought we knew about choline's impact on the cow and her calf, and top scientists have a lot to say about it. They are presenting new research that supports choline as a required nutrient to optimize milk production, choline as a required nutrient to support a healthy transition, choline as a required nutrient to improve calf health and growth, and choline as a required nutrient to increase colostrum quantity. This new research is solidifying choline's role as a required nutrient for essentially every cow, regardless of health status, milk production level, or body condition score. Learn more about the science that is changing the game and the choline source that is making it happen. Reassure precision-release choline from Balchem. Visit balchem.com/scientistsay to learn more.
Topic number one, why does fatty acid composition likely matter when we feed choline in other methyl donors? First evidence that choline was an essential nutrient in humans was derived by work performed by Charles Bess and Frederick Banting. The work of these researchers led to the discovery of insulin. They utilized diabetic dogs with their pancreas removed. What they observed in these dogs is that they developed severe fatty liver disease. Although I don't understand sort of the reason why they gave lecithin as a dietary treatment or as part of a component of the diet. Lecithin contains phosphatidylcholine, and when they gave lecithin to these dogs, they observed marked reductions in hepatic lipid composition. When they removed the lecithin, the liver lipid content was around 22% in the dog. But when they gave that lecithin to the dog, kept the lecithin in the diet, liver fat content was just 4%. I think this body of work initiated decades worth of research focused on the relationship between dietary choline, phosphatidylcholine in the cow, and as well as humans, and fatty liver progression. Flash forward to present day, fatty liver is a concern for fresh cows because of its relationship with ketosis, poor fertility, and compromised milk production. Our lab has routinely characterized the bovine lipidome to better develop nutritional approaches to prevent this disease. So in this figure here, we worked with a commercial dairy farm in southern Pennsylvania. Livers in cows were biopsied during the fresh period. We then analyzed over 300 lipids, not just phosphatidylcholine, but other lipids as well, in liver and plasma tissue. Cows were classified as having liver lipid content of less than 5%, so healthy animals, or high liver lipid content greater than 12%. The top features in plasma that reflected the fatty liver condition were low circulating concentrations of a complex lipid called phosphatidylcholine. What we noticed is that we detected over 100 phosphatidylcholines, but the majority of those that were low had a very low degree of saturation. They were rich in unsaturated fatty acids. Phosphatidylcholine is a membrane lipid composed of two fatty acids, and again, most of them can be unsaturated. The lipid has a metabolite within it called phosphocholine. This is a common storage form of choline, and it's a metabolite that's often responsive to rumen-protected choline feeding. So the take-home message here was that the development of fatty liver in cows develops with low circulating concentrations of phosphatidylcholine. We also observed something very similar in the liver. And I promise not to dwell too much on the past, but many studies have demonstrated the ability of rumen-protected choline feeding to lower liver triglyceride deposition. For instance, Hartwell and coworkers demonstrated this in overconditioned cows. Ellick and colleagues observed this effect with lower circulating BHB concentrations. And others have shown evidence for improved VLDL assembly with choline feeding. Now, this is particularly important when we talk about choline and fatty acid nutrition, right? Because rumen-protected choline feeding is believed to prevent fatty liver disease by enhancing the secretion of triglyceride within very low density lipoproteins, VLDL from liver. It does this because choline supports the hepatic synthesis of phosphatidylcholine. In fact, there are two key pathways for this. Right? The first pathway is referred to as the cytidine diphosphate choline pathway, CDP choline pathway. The initiation of this metabolic route relies on choline. In non-ruminants, the majority of phosphatidylcholine made is believed to be generated by this pathway. The second alternative route, often referred to as the compensatory pathway, is the phosphatidylethylamine methyltransferase pathway, abbreviated PEMT, which is an enzyme. It's an enzyme involved in transmethylation. It relies heavily on methyl groups, 3 of them exactly, to methylate phosphatidylethylamine and form phosphatidylcholine. These methyl groups can come from methyl donors such as methionine, choline, or betaine, but the process relies on folates and B vitamins as well. Phosphatidylcholine is important because it forms the monolayer of the VLDL particle that contains triglyceride within. Inadequate phosphatidylcholine synthesis may limit VLDL assembly and thus triglyceride secretion. Right? So choline feeding has the potential to prevent this. Rumen-protected choline feeding has been shown to increase phosphatidylcholines in VLDL fraction in plasma. We collaborated with the University of Florida, and the study involved feeding rumen-protected choline ion as reassure. For reference, 60 grams per day is equivalent to 13 grams per day of choline ion. In this particular study, we saw an increase in phosphatidylcholines within VLDL. We also observed increases in hepatic phosphatidylcholine concentrations, an increase in triglyceride within VLDL and plasma. And Zenabi and colleagues at the University of Florida demonstrated the ability of this practice to decrease liver triglyceride deposition. Now, this was in prepartum cows experiencing negative energy balance. So collectively here, data suggests that rumen-protected choline feeding can increase phosphatidylcholine synthesis to support VLDL triglyceride secretion from the liver to prevent fatty liver disease. So this brings me to why we should consider fatty acid nutrition when feeding methyl donors. First, the figure illustrates the 2 previous pathways I described. The CDP choline pathway in blue relies on choline, as you can see. You don't see any methionine there. In non-ruminants, this pathway utilizes diacetylglycerol rich in saturated and omega-6 fatty acids like linoleic. If the same is true in the transition cow, these fatty acids are coming from the diet, but more importantly, from adipose tissue mobilization. So there's an ample supply of fatty acids, I think, to support the CDP choline pathway in fresh cows. Right? The PEMT pathway is shown in red. This pathway relies on methyl donors, including choline, betaine, and methionine. It also utilizes phosphatidylethylamine formed from diacylglycerol that has a very low degree of saturation. In non-ruminants, this pathway prefers very long-chain polyunsaturated fatty acids, like DHA. However, in the transition cow, the proportion of these fatty acids relative to the total is at its lowest. I hypothesize that an inadequate post-ruminal PUFA supply likely limits PEMT activation, and thus you can have these methyl donors in the diet, like choline and methionine, but PEMT would remain inactive because of inadequate availability of unsaturated fatty acids. Okay. So we started to explore this possibility in dairy cows. In this study, we abmasally infused all cows with a high dose of a palmitic acid emulsion. So there was a lot of palmitic acid going post-ruminally. Cows were then abmasally infused with or without choline chloride. In panel A, choline infusion was able to increase or decrease plasma phosphatidylcholine concentrations, but this response varied based on the saturation of the phospholipid. In panel B, we showed that choline infusion increased saturated phosphatidylcholine like the one shown, which we proposed to be originating from the CDP choline pathway. But we actually observed decreases in PUFA-rich phosphatidylcholine like this one that contains DHA in panel C. Perhaps choline was unable to activate the PEMT pathway when saturated fatty acid supply was more than adequate. The proportion of DHA available to the cow was low. In the same study, we also infused cows with fish oil rich in DHA. We started to do this as well with different types of rumen-protected fish oil supplements. We observed increases in phosphatidylcholines containing DHA. Other phosphatidylcholines rich in PUFA that were not found in the fish oil were also responsive, suggesting that it's possible that that fish oil treatment was able to promote PEMT activation and just take up those other polyunsaturated fatty acids and to support phosphatidylcholine production. However, the same was not true for saturated phosphatidylcholine. We gave this fish oil, we did not see a change in those that were saturated. You know, in humans, DHA and choline dietary co-supplementation together works better than feeding only DHA or choline alone at activating PEMT. Right, so this is actually a pretty hot topic right now, looking at the synergy of these two nutrients. We're considering the possibility that increasing post-ruminal unsaturated fatty acid delivery, not just DHA, but other omega-3s like linolenic acid, but also MUFA like oleic acid, how might they help boost the activation of that PEMT pathway and support the ability of choline, methionine, or betaine to promote phosphatidylcholine production and alleviate fatty liver disease? You know, to show the ability of dietary fatty acid feeding to modulate choline supply, we recently fed lactating cows one of two prilled palm fat supplements. An MPA contains 72% palmitic acid and 12% oleic acid. HPA was pure palmitic acid at 99%. Feeding cows the palm fat containing oleic acid increased total phosphatidylcholine, total sphingomyelin, and total choline supply relative to cows that were fed the palm fat containing only palmitic acid. So I know this doesn't test the PEMT hypothesis, but it does show that depending on the dietary fatty acid composition, we could potentially influence endogenous choline status. So I think this deserves further consideration moving forward, especially for fresh cows that are fed commercial fats that are high in saturated fatty acids. So to end, for conclusion one, low phosphatidylcholine supply is a key feature of fatty liver in dairy cows. Right? It's likely due to low PUFA and choline supply, that definitely in situations where post-ruminal availability of PUFA and choline are limiting. Delivery of PUFA, protected PUFA, that are protected from rumen degradation could potentially support phosphatidylcholine synthesis in the cow. We should consider the interactions, though, with fatty acid digestibility, which I'm not going to get into at the moment. The other added benefit of PUFA is that there's an improvement of insulin sensitivity. That improvement in insulin sensitivity has the potential to reduce body fat mobilization as well as inflammation. And a reduction in body fat mobilization can further decrease the hepatic fatty acid uptake and thus also provide an additional mechanism to prevent fatty liver. So here are some fresh cow considerations. I gave a talk last week on nutrient partitioning and the role of fatty acids in the diet. We should be keeping dietary fat 4 to 6.5%. When we think about commercial fats, we're starting to see a lot of these different types of commercial fats that are available. They can be mixed with unsaturated fatty acids. It's important to ensure, though, that any addition of commercial fats to the diet, typically at 1/3 of total dietary fat, that those unsaturated fatty acids are protected. For that talk, I went through the literature and started studying the ratios of saturated fatty acids, unsaturated fatty acids in the diet. It's pretty clear that as you get closer to 1-to-1 proportion of saturated fat to unsaturated fat in the diet, you're going to increase nutrient partitioning to the mammary gland, but potentially accelerate body condition loss, and that could exacerbate the fatty liver condition. Looking at the omega-6 to omega-3 fatty acid ratio, less than 5 in fresh cows is best. Trying to increase the incorporation of omega-3 fatty acids like linolenic acid. And certainly linolenic acid likely has similar effects on PEMT pathway and choline and methionine efficacy, but fish oil is certainly going to be more effective at lower doses. Feeding 12.5 to 20 grams per day of rumen-protected choline ion and keeping metabolizable methionine high in fresh cow diets will also ensure that we're providing all the necessary components for the pathways that generate phosphatidylcholine in the liver. Topic number 2, choline degradation is something that we want to avoid. Okay, this is why rumen-protected supplements were developed to prevent choline degradation in the rumen. The next few slides will describe new data obtained by our lab that suggests that choline degradation may also occur post-ruminally and thus limit bioavailability. So in the gastrointestinal tract, choline but also carnitine, choline and carnitine together, are metabolized by gut bacteria. And this figure actually comes from a biomedical research paper focused on human nutrition. But in non-ruminants, it's fairly well established that in the small intestine, choline and carnitine can be converted to a metabolite called trimethylamine by an enzyme called trimethylamine lyase. Now, not all bacteria, as far as I know, have this trimethylamine lyase. Right? Different bacterial populations will express it. But that trimethylamine lyase can convert choline to trimethylamine, TMA. That TMA in non-ruminants is converted to a metabolite called trimethylamine oxide in the liver, abbreviated frequently as TMAO. And that's done by an enzyme called flavin monooxygenase. That TMAO can be absorbed by tissues once it's released from liver, or excreted in urine, or in the cow, or even the postnatal woman, it could be found in milk. At its core, TMAO is a marker of gut choline degradation because it can only come from the formation of trimethylamine by gut bacteria. So what about the cow? Well, I'm sure at least a few of you are aware of this table. Unfortunately, I forgot to add this additional site. This is from Sharma and Erdman in 1988, I believe. They were looking at feeding choline, unprotected choline, and looking at rumen choline degradation. They fed rumen unprotected choline at low and high levels, from 24 to 326 grams per day. That's crazy high. The flow of choline through the duodenum was only slightly responsive, increasing from 1 to 2.5 grams per day. The authors evaluated ruminal choline degradation, and it exceeded 97% when choline was unprotected. Nearly all of it was degraded. So this is why we have protection. Well, the authors did not measure trimethylamine or TMAO, but they did reference previous papers. Research as early as 1959 has been demonstrated that the addition of choline to in vitro rumen fluid cultures resulted in the marked degradation of choline, but increases trimethylamine. That's what you see in panel B. This would be caused by the actions of trimethylamine lyase. Others have investigated the fate of trimethylamine, what happens to it. Well, during fasting in ruminants, that trimethylamine is rapidly converted to methane. Data suggests that during feeding, the conversion of methane decreases substantially, meaning that trimethylamine may pass the rumen. Collectively, these data confirm that unprotected choline can be converted to trimethylamine in the rumen, which may be available to the cow it's fed. The implications also extend to choline in more complex forms. We recently completed a lecithin feeding trial, and lecithin contains, in part, phosphatidylcholine. Right, cows were fed D-oil soy lecithin at 0 to 0.36% of ration dry matter. Our fatty acid data suggested that lecithin was extensively degraded in the rumen. I'm not showing it here. That phosphocholine was released. In support, we observed marked increases in circulating TMAO concentrations. This TMAO had to have originated from choline degradation and trimethylamine formation in the gastrointestinal tract. Can't say conclusively whether or not that was from the rumen or the intestine. Up until this point, I've discussed ruminal choline degradation, right? But that same abmacyl infusion trial that I described earlier, where we abmacyl infused choline chloride in the unprotected form to lactating cows, we observed marked increases in plasma choline concentrations like we would expect. Choline metabolites like betaine and dimethylglycine were also responsive to choline infusion. That also makes sense because when you feed rumen-protected choline, you may not see changes in free choline concentrations, but those choline metabolites, betaine and dimethylglycine, are often responsive. But we were surprised to see an increase in plasma TMAO concentrations. These data suggest that choline was degraded post-ruminally to trimethylamine by bacteria. We can say this because we abmacyl infused the choline chloride, and that trimethylamine was subsequently converted to TMAO in the cow's liver. This is certainly not a desired response because our hope is that post-ruminal choline is reserved for intestinal absorption only and is not degraded. Recent work, however, by Dr. Deveth, not presented here, suggests that the bioavailability of choline that enters the abomasum is not 100%. It's possible that some of that choline is degraded to trimethylamine. We just don't know the exact amount quite yet. So the one concern is that choline is degraded in the intestine and thus limits its absorption. Okay, that's certainly an issue. At the same time, however, my lab knew very little about TMAO, and so I had my graduate students do a PubMed search, started reading the literature, and it was enlightening. Soon after the reports of trimethylamine in cows, research focused on TMAO, different studies, have increased in the literature substantially. And I think it's considered a relatively hot topic at the moment. The reason is that TMAO is associated with poor health in rodents and humans. For instance, non-alcoholic fatty liver disease, inflammation, insulin resistance, obesity, oxidative stress, and cardiovascular disease are all associated with the accumulation of TMAO in plasma. More recent studies have aimed to determine whether TMAO causes these poor health outcomes, because for the longest time, these were clearly just associations. In some cases, this has been modestly proven that TMAO can cause health impairment. The strongest case is for the ability of TMAO to promote oxidative stress and cardiovascular disease, and this is predominantly in rodent models. There are also some studies demonstrating the ability of TMAO to promote fatty liver and diabetes, right? Insulin resistance, again, in rodent models. This is certainly alarming to us because when you think about it, cow has fatty liver disease, you're feeding rumen-protected choline to prevent this, right? And severe insulin resistance could accelerate body fat mobilization in cows. But we certainly recognize that rumen-protected choline feeding has consistently proven to have a positive effect on health. Can't ignore that. But we were certainly cognizant that not all transition cow studies see a triglyceride lowering effect with rumen-protected choline feeding. I think that might relate to the fatty acid feeding in the diet, but we've considered TMAO as well. So we searched the literature again, and we did find a single metabolomics paper demonstrating a positive correlation between circulating TMAO and fatty liver in transition cows. All right, so this had us a little bit nervous. So we recently completed a study to explore the effects of TMAO on milk production and health in early lactation cows. In a replicated 4x4 Latin square design, 8 cows were continuously intravenously infused TMAO at one of 4 doses, 0 to 60 grams per day for 6 days. We collected plasma, liver, milk, and urine during each experimental period. In panel A, we clearly demonstrate the ability of this approach to increase plasma TMAO concentrations in a stepwise manner. As expected, we observed marked increases in urinary and milk TMAO concentrations. The good news is that urine is the origin of disposal and not milk, just by comparing the concentrations. To date, we've actually quantified TMAO yield in milk, and even at these high doses, the yield is not going to exceed 100 milligrams per total day, per day in milk. At rumen-protected choline feeding levels in the field, I suspect that it's less than 10 mgs per day. That's total milk volume. So it's not a lot. So we wanted to consider that, though, considering the negative implications of TMAO on human health. Now, we're currently quantifying, though, the yield of TMAO in urine, but I think most of it's going to the urine. We then studied the effects of acute intravenous TMAO infusion on milk production. There was no changes in dry matter intake in these early lactation cows, no changes in energy intake, no change in milk yield. Yields of fat, protein, and lactose did not change. Milk content, fat, protein, or lactose did not change. Fat-corrected milk yield, energy-corrected milk yield, or the efficiency to produce milk did not change with treatment. And so, granted, this is an acute intravenous TMAO infusion. These animals were not fresh cows, but it appears as though that TMAO does not influence milk production. We performed glucose challenges to evaluate glucose utilization and insulin response in these early lactation cows treated with or without TMAO. Recall that TMAO accumulation causes insulin resistance in rodents. And we don't want that to happen in postpartum cows. Although we did not measure insulin, the good news is that we did not see a change in the glucose response. Because insulin suppresses adipose tissue mobilization, we studied circulating fatty acids. In response to that glucose challenge, we did not detect a difference with treatment. We also measured plasma glucose, triglyceride, and total fatty acids every day of the experimental period in samples collected before feeding. We did not see a treatment response. We performed a serum liver panel to look at different liver enzymes that we could better understand hepatic health in these animals that were given TMAO. We did not see any change with treatment. And we lastly, just to be safe, we analyzed over 50 polar metabolites using metabolomics and over 300 lipids using untargeted lipidomics, a fully comprehensive approach using mass spectrometry. And for the most part, we observed very little change. So, second conclusion, trimethylamine oxide does not appear to influence energy metabolism or challenge health in early lactation cows. We really needed to address that question because certainly when you feed rumen-protected choline, the goal is to improve health in milk production. So we're moving on from this. We do not think TMAO is causing any kind of health impairment or impairment on milk production. But we still have a challenge ahead of us, and that's bullet number 2. Choline is subjected to ruminal, but also lower gut degradation to trimethylamine. We've shown this with the avomacil infusion of choline chloride. The influence of this lower gut choline degradation on choline bioavailability needs to be defined. I will say, though, we're starting to think about how might diet influence that microbiome, whether it be ruminal or even lower gut microbiome, and thus influence choline degradation to trimethylamine. Data in non-ruminants suggests that unsaturated fatty acid feeding is a means to shift the gut microbiota and slow trimethylamine formation and improve choline bioavailability. So this ties in nicely with the fatty acid feeding recommended earlier. Post-ruminal availability of PUFA has the potential, we need to test this, to reduce choline degradation and increase choline bioavailability. All right, final topic. Topic number 3. A few of you may know that I have this sort of interest studying lecithin, phospholipids, and fatty acid digestibility. Well, lysophosphatidylcholine is a component of lysolecithin. All right, looking at the structure of lysophosphatidylcholine shown there, it uses a single fatty acid, all right, in its production. Right? In ruminants, the most abundant lysophosphatidylcholines include those that utilize palmitic, stearic, and oleic acids. Right? Lysophosphatidylcholine also contains phosphocholine. All right, and plenty of data have shown that rumen-protected choline feeding is able to increase lysophosphatidylcholine concentrations. It's one of those choline metabolites that's responsive to choline feeding. Well, this topic focuses on how lysophosphatidylcholines may modulate immune function in dairy cattle. But first, I wanted to provide some background about immunity. Recall that cows, and as humans as well, have innate and adaptive immune systems. Adaptive immunity is specific for pathogens, but slower to act. The innate immune system is fast-acting, but non-specific, and composed of different cells like neutrophils. Activation of neutrophils involves something called the oxidative burst, which is the rapid release of reactive oxygen species, including superoxide anion and hydrogen peroxide. This involves the enzyme NADPH oxidase. This oxidative burst contributes to pathogen killing, but the process also involves low pH and enzymes found in the phagolysosome. Why is this important? Well, the neonatal calf relies on passive immunity from colostrum, but this immunity fades. And because innate and adaptive immunity are underdeveloped pre-weaning, the calf experiences what is referred to as a gap in immunity. In panel A, these data demonstrate that the ability of neutrophils to elicit the oxidative burst and promote phagocytosis is diminished pre-weaning. We also like to talk about immunosuppression in transition cows. In panel B, the oxidative burst is diminished in multiparous cows and declines with time in primiparous cattle. So researchers have sort of been on the hunt here for dietary approaches to bolster immune function in pre-weaned calves and transition cows. Dr. Bradford at MSU is certainly the immunity guru. He recently had a study where he evaluated the effects of choline supplementation on bovine neutrophils and monocytes. In brief, he observed that the phagocytic and killing ability of isolated neutrophils were linear diminished with increasing doses of choline. He detected decreases in oxidative burst and pro-inflammatory TNF-alpha. He also had provided evidence that there was an increase in choline metabolism with this choline supplementation. Now, the conclusions were a bit broad because it's a bit early to make more definitive conclusions, but it was clear that choline was able to modulate neutrophil immune function. My interest in lysophosphatidylcholine actually stemmed from a collaboration I had with Dr. Baumgart. He gave endotoxin, lipopolysaccharide, to some lactating calves that were experiencing hyperlipidemia, high circulating fatty acids. He saw the typical response, an increase in rectal temperature, an increase in circulating fatty acids in response to the hyperlipidemia induction protocol he had. He infused them with triglyceride to try to mimic the fatty liver conditions that you would see in a fresh. Well, what was important here is in panel C that when he gave the endotoxin, there was the activation of the acute phase response, an increase in serum amyloid A. He also saw changes in LPS binding protein. These animals were experiencing an infection, pathogen infection, and were responding accordingly. Well, we took these samples. We took all the plasma samples from all the time points and liver tissue, did our omic work, and long story short, we noticed marked reductions in lysophosphatidylcholines. I'm sure this would happen. In lysophosphatidylcholines. Now, this decrease was about 80% decrease by hour 16, but the decrease in lysophosphatidylcholines occurred as early as hour 4. And in transition cows, they're experiencing that immunosuppression. They also have low levels of lysophosphatidylcholine right at parturition. So for now, we've established this relationship between endotoxemia, immunosuppression, and lysophosphatidylcholine status, that LPCs are low at this time. So we got back into the literature because we didn't have an hypothesis up until this point. But in the literature, people that die from a bacterial infection, sepsis, also have low lysophosphatidylcholine concentrations. And there have been plenty of studies looking at lysophosphatidylcholine therapy as a way to protect against a bacterial infection. This is just one study. They gave, basically they used this approach where they use what's called a cecal ligation procedure. They open up the abdomen, they puncture the gut, they let the bacteria enter the gut, and they close the gut, and then eventually the mice are going to die. They'll basically all die within 10 days. But if you gave those same mice lysophosphatidylcholine at the start, mortality was prevented. And at the high dose here, it was nearly almost completely prevented by day 10. And in panel B, they also subjected mice to pure E. coli injections, live E. coli. Certainly that is going to proliferate in the animal, but the addition of lysophosphatidylcholine was able to increase E. coli killing. Data looking at the mechanisms has identified the ability of lysophosphatidylcholine to activate neutrophils. Right? It promotes the oxidative burst. It can suppress long-term inflammation in response to a bacterial infection. It can clear out E. coli faster. It also has been shown to help bolster adaptive immunity. It can increase immunoglobulin production and interferon gamma availability. So we took the opportunity. We just started a series of experiments looking at lysophosphatidylcholine therapy in diet as a way to help boost immunity in animals that are experiencing immunosuppression. And so here we had pre-weaned calves. We isolated some neutrophils from pre-weaned calves. I think this is around week 2, week 3 of life. And we cultured those neutrophils with lysophosphatidylcholine. And we had 2 types here, palmitic and stearic. We've also done this with the oleic acid version. And what we observed is that saturated lysophosphatidylcholine was able to enhance the oxidative burst of these bovine neutrophils. So we gave PMA. PMA is 4-bomb-myristate acetate, and what it does is it activates the neutrophils, promotes the oxidative burst, but the presence of the lysophosphatidylcholines amplified this response at increasing doses. Well, just to remind you, rumen-protected choline feeding is able to increase lysophosphatidylcholine supply. We're currently considering how not just choline feeding might influence immunity, right, especially in transition cows, but looking at those emulsifiers, lecithin and lysolecithin. So to conclude here, for topic number 3, immunosuppression and endotoxemia are characterized by low circulating lysophosphatidylcholine concentrations in dairy cows. In vitro data suggests that saturated lysophosphatidylcholines activate neutrophils, and that rumen-protected choline feeding is able to increase lysophosphatidylcholine. Dr. Bradford has shown that free choline is able to modulate immune function of neutrophils. So there's certainly a connection here that's between immunity and choline status and the involvement of lysophosphatidylcholine. As we start to look more at this work with lecithin and lysolecithin, I fully suspect more of these kinds of studies in the future. Please be aware that that lecithin does have phosphatidylcholine, which can be degraded to lysophosphatidylcholine and absorbed, but it's not the only phospholipid found in lecithin. And the same thing with lysolecithin, that it does not just have phosphatidylcholine. In fact, crude forms of these will also have triglycerides, which is going to have different response on immunity, obviously. One last thing, some of our speakers here, Dr. White, I'll point her out here, but we had a bunch of collaborative authors put out this review paper, 1-carbon metabolism and methyl donor nutrition in a dairy cow. I think it's a decent summary of the current state of the literature. You can catch this in JDS. Thanks to our collaborators, the late Dr. Staples, Dr. Baumgard, and Dr. Howie at Johns Hopkins.
The economics of feeding Reassure precision-release choline. Reassure is fed during the transition period, and because it's fed for such a short period of time, it costs just $15 per cow, and yet the benefits will continue to generate income throughout the year. Cows fed Reassure produce 5 pounds more colostrum, which pays for your Reassure investment on the very first day of lactation. Cows fed Reassure also produce 5 pounds more milk per day, every day. That means after the first day, every day is payday. Invest in Reassure during the transition period and recoup your investment on the very first day of lactation. After that, you got it, payday.
So we have quite a lot of questions that have come in from our audience, so let's get right into those. First one, can milk TMAO be considered a marker for choline degradation?
Yeah, so I mean, this is something I'm looking into. I think TMAO is a marker, whether it's plasma or milk, could certainly be a marker of choline degradation. But I want to be careful here because we don't necessarily know where that TMAO is coming from. We don't know if it's coming from the intestines or the rumen quite yet. So I think if you're thinking about rumen-protected choline supplements that have a high degree of rumen protection and that most of it is being provided to the intestine, then we could certainly make assumptions that what you see in milk could be majority derived from intestinal degradation. I also think it's just a possible means to compare supplements. But I think you have to compare it within the context of bioavailability. And so that requires a little bit of intricate trial design to do. Yes, it's certainly possible.
This one's a little complex. So lecithins are reasonably effective surfactant, and there's literature out there relating surfactant feeding to changes in nutrient absorption. So to what extent have you been able to separate shifts in nutrient supply at the lumen wall from a more direct mechanistic effect of the lecithin molecule?
Yeah, I mean, it's a very complicated question. Probably requires a decade's worth of research. I mean, you're absolutely right that, it's going to help in emulsification in the lower gut. It's certainly going to have consequences on perhaps fatty acid absorption, which could also influence immune function. So I think it's too early to definitively point at lysophosphatidylcholine as the certainly endogenous supply of lysophosphatidylcholine as the sole immunomodulator. There's certainly other implications. That's why I talked about the fatty acid digestibility component, because certainly increasing fatty acid supply could have benefits on immune function and health of the animal. Now, in terms of I think there was a question there about the availability of other nutrients. Now, it's interesting because I'm not sure if this is what you're asking, but choline within lysophosphatidylcholine, there's some evidence to suggest that the bioavailability of choline within lysolecithin is greater than the bioavailability of unprotected free choline chloride, free choline within choline chloride. Now, that's all based off of just a couple of papers in non-ruminants, and one including human paper. Now, that being said, the amount of choline within lysolecithin is substantially less than you might be feeding in a rumen-protected choline form. But if the bioavailability is greater, perhaps they're near equivalent. I mean, that's just a pure guess at this point, but hopefully I answered part of the question there.
All right, so let's, you mentioned undigested fatty acids a moment ago, and this question was about those. Getting lost somewhere in the relation between UFAs and choline. So requested if you could elaborate on that relationship a little.
Yep. So I'm thinking about it in the liver. That's the key take-home message here, that if you can provide more polyunsaturated fatty acids, particularly omega-3 fatty acids to the liver, to the cow, then you could support phosphatidylcholine production. One of my arguments here is that you could be feeding methionine, choline, methyl donors in general, and one of the intended outcomes is to activate methylation to produce phosphatidylcholine. But you might not be able to do that, particularly in a fresh cow that has limited endogenous supply of PUFA to begin with. And so if we're able to increase the post-ruminal availability of PUFA that are protected from rumen degradation, then we could potentially support the ability of methyl donors to generate phosphatidylcholine and prevent fatty liver disease.
All right, so along those lines, would it be possible to isotopically label choline to look at TMAO in milk or plasma to measure a rate of degradation?
Yeah, absolutely. I think maybe you're my graduate student. We're exploring that, yes.
All right, one of your colleagues has another question here about the connection between choline incubation and LPC incubation. So why did LPC increase the oxidative burst of neutrophils, but choline decreased it?
Yep. All right. So, the thing with the choline is that, first of all, there are two different metabolites, for one. And choline most likely is getting taken up by those cells, being metabolized inside the neutrophils. The way the lysophosphatidylcholine acts is that it binds to receptors on the plasma membrane, right? And so there's two different modes of action there. And so choline is not going to be able to interact with the lysophosphatidylcholine receptor. Now, I also will say, too, that the effects that they're seeing with the choline in those in vitro cultures, you might not see that in vivo in response to rumen-protected choline feeding, because often you do not see a free choline response with feeding rumen-protected choline. You will see the lysophosphatidylcholine response. So I think we just don't have enough data there to sort of distinguish that, but there's just sort of something to be aware of.
All right, Dr. McFadden, that's where we're going to have to end it. Great answers, and thank you to all of our attendees for those questions.
We'd love to hear your comments or ideas for topics and guests, so please reach out via email at anh.marketing@balchem.com with any suggestions, and we'll work hard to add them to the schedule. Don't forget to leave a 5-star rating on your way out. You can request your Real Science Exchange t-shirt in just a few easy steps. Just like or subscribe to the Real Science Exchange and send us a screenshot along with your address and t-shirt size to anh.marketing@balchem.com. Balchem's Real Science Lecture Series of webinars takes place on the first Tuesday of every month, with the top research and nutrition topics that will impact your business. We also include small ruminant, monogastric, and companion animal-focused topics throughout the year. Visit balchem.com/realscience to see the upcoming topics and to register for future webinars. You can also access past webinars and search for the topics most important to you.